Okay, so let's have a class, and today we will do a review for the exam 1. Exam 1 will be Thursday, same time here until 12:15. Okay, if you have a university accommodation request or all kinds of requests, you just find me in the office around 10:30 in the morning. Then I'll give you a separate room to do it. Okay, we have a testing room in our building. I'll give you a room to do it separately. Honestly, you don't have to do that. And if you really needed it, you can do it, and the extra time it could be offered you if you need it. So now, how are you going to prepare? First of all, you don't need to be panicked. You're going to study very well for these practice exams. Now, how you study? It's not like you're going to have the question, number 37, it's B. Then Thursday early in the morning you see 37, or it's B. Or 38, it's A. Or 39, it's A. Or then you go to the classroom. When you prepare these questions, at the same time, you open your slides or the class notes. Then you take the notes or highlight the area which is on your slides, what we talked about already, and why that question related to the exam question, what's the knowledge we can use. This is the way we're using the practice exam. So, lots of you ask a question for when you're going to post on the key. I will post the key after today's class. These couple past days, which is let you to study by yourself, to find the answer, and then use the time to study. That's also the way to practice, and also the way to review the questions. Okay, that's why we give you the practice questions. Then I will talk about basically one by one, but if I don't have time, some of them you can just look at the answer. But the key points we absolutely will mention. I think this whole exam practice questions, oh, by the way, the real exam questions will be very similar, the same level. Okay, it's not difficult. Which means if you can close the book right now, just pick and get 100, then you will be 100 in the exam for sure. There's no problem. Some of these questions is relatively difficult, so I'm going to talk about first. And then I'll show you the way what we should supposed to be do the review. Let's look at question 37. We will going to do in the lab. So I'm very glad that today our lab is doing Macanthi agar, mannitol salt agar, blood agar, and introduced to chocolate agar. So let's look at this question, because this is not in our slides, but we practiced we're going to do in the lab. So mannitol salt agar only allows the growth of halophiles, salt-loving microbes. Non-halophiles will not grow. Among the halophiles, mannitol fermenters will produce acid that turns the pH indicator yellow. Mannitol non-fermenter leave the medium red. Onto MAS, you inoculate a halophilic mannitol fermenter and a halophilic mannitol non-fermenter. In this case, this media is acting as what? Okay, lots of you will do. It's not even read it. Because you said mannitol salt agar is a selective media, is a differentiated media, so I pick C. Then finally you find it's wrong, then you go to my office and say, oh, why it's wrong, talk about the whole day. What's the mistake you made? You didn't read. So the first thing in the exam is very important. Read. Okay, read the question carefully, what they are talking about, not just in your mind. Oh, mannitol salt agar is a selective and differentiated media, so I pick C in 5 seconds. So read the question. So let's talk about that. Then we can save a little bit of time in the lab. Okay, mannitol salt agar. There are two ingredients. It's mannitol and salt. Obviously, they have some function. What is salt? It's about like 1 to 3%. We call it osmotic balance of bacterial cells. So some of the bacteria doesn't like it. Some of the bacteria like it. Let's say Staphylococcus aureus can grow on the skin surfaces. Okay, so that's why we call it a halophile. And we give them a specific name. So for example, E. coli cannot grow. So we call it a non-halophile. That's a selective reagent, is salt. Now what's a mannitol? That's 6-color sugar. This is used for the energy resource, but the more important, it's used during the fermentation. We'll talk about fermentation detail in exam 3. Because they're going through fermentation, there are 2 ways, which means they're used or not used. If they use the fermentation, which means mannitol going through fermentation, it will generate an acid. Because fermentation at the end of the day is acid, like the cheese, little acid, like lactic acid based drink, it's little acid. Like your smoothie, even, a little acid. Now, mannitol fermentation, not positive, of course, it's no acid. Now, once it's generated acid, what means? The pH is going to decrease. Once the pH decreases, there is a pH indicator there called phenol red. So, the colony looks like it is, the color is yellow. And obviously, this is nothing going to happen. So, it stays the same. Stays the original color. So what this tells us, the function of this media is differentiate and selective. Okay, now we go back to look at the question. That's a background. What this tells you. This is on to MSA agar. So here is just what I said. Introduce you. Okay, give you a story, what is mannitol salt agar. Now on to MSA, you introduce a halophilic mannitol fermenter and a halophilic mannitol non-fermenter. First of all, are those 2 colonies going to grow? Yes, those 2 different bacteria will grow because both are halophilic. So this guy at this moment doesn't really help too much. Because both of them like salt, so they're going to grow. Now how do we know which one is which? Because the color is different. One is mannitol fermentation positive, another one is negative. So what is this telling us? This media is acting as a differentiated media. Because the selective reagent, salt, did not do anything. Okay, so the answer is B, differentiate media. Okay, so that's one example I want to mention first. The second example is always this mess. So question number 42. Let's do that one. Given a log phase bacterial culture with 1 multiple 10 to the power of 6 cells per ml, and the generation time is 30 minutes. How long does it take the culture to reach a density of 6.4 multiplied by 10 to the 7 cells per ml? Okay, when you do this question, you may be struggling with the maths, may be confusing. But let's do this one first. What do we call the review? Very important on your mind. Think about what the bacterial growth curve. Okay, x-axis is time. Y-axis is population. Or you can simply put a number there. Now what's the growth curve looks like? This, this, this. Therefore, you know, lag phase, log phase, stationary phase, and this phase. Okay, what we talk about here, why this question will generate? Because bacteria grows dramatically after they accustom to the environment, going to log phase. So there is a question comes up. Originally, let's say around here is 10 to the power of 6. After certain time, it grows to 6.4 multiplied by 10 of 7. Okay, first thing, that's a power of 6. That's power of 7. It's confusing. So let's make it the same. 6.4 multiplied by 10 to the 6. Okay, so this gives you what? Which means how many generations you need. Okay, this gives you 10, 64. Can you do it? If you don't know how to do it, then that's what I said. You need to list all these things in your mind or in your draft paper, whatever it is. Is that right? So n equals what? 6. What this means? 6 generations. Because for the bacteria we talk about, doubling time equals generation time. And how many generations here? 6 generations. What is the generation time here? 30 minutes. That's a bacteria doesn't grow too fast. 30 minutes equals 0.5 hour. Okay, then how we do it? Multiple 3 hours. Is that good? So this question is studied. The generation time is a concept. Studied the bacterial growth curve. So in the exam, if I ask you to draw a bacterial growth curve, then you should know how to do it. And then what you should do, then you go back to your slides. That's always how we should study. Okay, you go back to your slides. It's around roughly is lecture 4 PPT. Okay, then you turn it on. And you should read here what is called generation time. This is a complicated math. Maybe we don't test you. Is that clear? So these slides, these 3 slides, mark it, relate to question 42. That's what you study. Okay? So these are the 2 questions. Okay, now let's do another review. During the exam, we will ask you to label. So there is a very good question here is for you to label. Question 34. Label the diagram shown below. Clearly label the terms that apply. Note not all of the terms apply. Okay, which means you can just using the strategy which is when showing up, then you remove it, delete it. So let's say you have A, B, C, D, E, F. A is already picked, so gone. Or B already picked, gone. Then left F, then you put it there. No, that methods, that approaches may not be working. Okay, so that's what tells you. Terms, these could possibly be using gram positive, gram negative, outer membrane, inner membrane, blah, blah, all of these. We all talked about already. Now you look at this. Okay. First of all, what is the bacteria there? Gram positive or gram negative? Gram negative, yes. If you don't know where you should go, if you still look at it's confusing, then by the time, what you should do, is go back. You can see here, when we talk about the gram stand, okay? But more important, you're gonna go lecture 3 slides. Is right here. The gram positive cell was and gram negative cell was. So these 2 slides, you mark it related to question 34. Okay, now can you label? Of course you can label all these. Gram negative, what are these? That's called the fluid mosaic model. Is that right? That is called the peptidoglycan. That is outer membrane. That is lipopolysaccharides. Okay, now in your mind it looks like separately. But how are we going to study? We always study, go back to talk about the gram stand. That's I always say. It is important in your mind, there is some knowledge you can connect it, like a networking system in your mind. So, the key review of this section is we start to talk about the gram stand. And we did already a lot in the lab, we talked a lot. So Chris, the gram stand. First of all, 4 steps. Crystal violet, the basic dye. We have a multiple choice question for that. Followed by gram iodine. Let it stay strong, counterstand with 95% alcohol. Decoloration, counterstand with saffron, sorry. Okay. You should be very clear with this function, and this is more important. Okay. We have a question for that. That is in your mind. Then we talk about Y stays purple, Y stays pink. We talk about 2 theory. Number 1 is cell wall theory. Number 2 is lipid theory. Now this related to peptidoglycan, and this related to lipopolysaccharides. Okay, now in your mind, we're going to do this question. Then we do this. This is going to be very thin. Then you draw it. Okay, on the top, you draw it again. That's the same thing. So you can do it roughly, you can draw it carefully, that's okay. So on your notebook, you draw it. It took you 5 minutes, so 1 minute. First thing, don't forget, this is gram-negative bacteria. Then we label. What is this called? Cell membrane, is that right? What's the name for that? Remember we have the question in the field in the black? Mosaic model. By the way, I don't mean anything. If you sit here, you don't even know this is called a fluid mosaic model, then I would suggest you to read Biology 101, do you amateur time. Oh, okay, I have, because we had 140 students, I had like 3 or 5, some people are really not strong foundation of the knowledge. If you don't know this, then you go back to read your older textbook. Because these are the knowledge you should know. Okay? So for the mosaic model, we have that question. Then you go here, what's this transmembrane protein? Okay, then transmembrane protein. Okay, what is this guy called? Phospholipids, is that right? Then you can just say phospholipids. That's okay. I'm not that picky. Phospholipids is okay. What is this one? The head. So remember we talked about the chemicals. The head looks like it's ethanol, modified ethanol dissolved into water. Hydrophilic. How about here? Hydrophobic, which is lipophilic. Okay, you can do that. What is that? Peptidoglycan is very simple. Now you go ahead to study what we talk about the peptidoglycan. It's a modified sugar cross-linked with amino acids. So what are they? That's NAG, N-acetylglucosamine, and acetylmuramic acid. You don't need to worry about chemistry structure. Okay, we don't test you for that. Okay, but it's very thin. Now on the top, ultramembrane. Okay, what on the top? Lipopolysaccharides. It's LPS. LPS generates what? Endotoxin. Then what else you do? Go ahead to label gram-positive bacteria. It's the same thing. This is a major outline of the class what we talk about. So in your mind, you go into the classroom Thursday, you remember this. So that's a label. Okay, I think I have the key. I'll have a printed key for you. You can look at how you're going to label this. But keep in mind, not all the terms you're going to use. Okay? Okay, now let's go back to see some of the easy questions. First of all, we talk about this staining, the gram stain. That means, okay, by the way, don't talk there. Don't chat because I want everybody to get it. You know, I know you know, some people you know, some people doesn't know. So I want everybody to do very well in the exam. Okay, so don't chat there. You have a question, you can ask. So because some people maybe not even review yet. So I want to help everybody. Here we have the staining. Relatively complicated. And we have a question related to the staining technology. So we go back to see what are those questions for. Then you can use these to review. So we go back, all these. Okay, at the beginning, this is history. We talk about it later. That's relatively easy. These are microscope. Relatively not that difficult, so we go here. Okay. Now let's talk about some question for gram stain. Question number 17. This is a scenario happening in the lab all the time, and we mentioned it. If the decolorizing is left on too long in the gram stain procedure, gram-positive organisms will be stained blank, and the gram-negative organisms will stain. What this means? This step is staining too long. So you can think about when you finish here, and your boyfriend or girlfriend called you to say, I need you to leave. Then you come back, 2 hours, what happened? Then everything is blank, is that right? Then you can't stain with saffron. What this means? It's all pink. So you use those extreme situations could help you to understand. So what's the answer? It will be D, pink and pink. That's one of the example of a gram variable stain, which means even it's gram positive, it could staining like gram negative. Then think about in your mind what the other situation will be. Oh, the culture, is that right? Because peptidoglycan is already cracking down. So 2 scenarios we'll have gram variable stain is older culture, and this is a good example. Okay, then let's look at the others. 18. basic dyes such as methylene blue binding to cellular molecules that are. Okay, this is English, which means cellular molecules that are. So the dye is positively charged, and the cellular molecules are negatively charged. This is easy. Okay, it's a very basic, simple concept for the simple stain how we did. So the answer is B. Okay, so let's look at other things. Do we still have any other? So, question 21 is certainly answered here. Is that right? So question 21 certainly answered here. The basic dye is mordant to let the crystal value stay strong. Decoration accomplishment with saffron. So that's question 21. Let's look at do we have any other questions? Okay, question 29. On the top, lipopolysaccharides, very typical for gram-negative bacteria. Lipopolysaccharides has lipids A, so that fits lipids here. They generate endotoxin. And the endotoxin, we said, is heat liable, which means cooking can solve the problem. That's a very easy question. The lipopolysaccharides that is found in the outer membrane of gram-negative bacteria is also known as endotoxin. Okay. Exotoxin, we haven't talked about it. Murine, we didn't talk about it. Where is teichoic acid then? That's a gram-positive bacteria on the cell wall. That's very good. That's what you're going to study. Okay? You don't just go early in the morning, have your bread, say, oh, this is D. Oh, then it's D. Then you go back ask yourself, what is teichoic acid? Where it is. If you don't know, let's say you don't know, go back to see the slides there. T-E-I-C-H-O-I-C, teichoic acids. Okay? That's how we study it. Okay, that question is gone. Then let's see other questions. I don't think we have others, but we have some questions related to the staining technology. So we want to talk about that. Let's look at question 15. Prior to staining, smears of microorganisms are heat fixing in order to. And I said heat fixing is important. What that means? Ensure, remove the dust particles from slide surface? No. Attach firmly. Allow eventual visualization, internal structure, create small pores. So what that means? The answer is C. Because we are inactivate the enzymes. Basically you kill it, so attach it firmly to the slide. Okay, acid-fast microorganism. Mycobacterial tuberculosis contains, because of mycoric acid, it's a very waxy cell wall. So mycoric acid is what? Is an unsaturated fatty acid, it's a long chain. So it's belonging to what? Lipids. Now let's say you don't know. What we should do is go back to the very beginning of lecture 2. When we talk about the acid-fast stain, we didn't practice in the lab, but we talked about right here. They gave you the example is mycobacterial tuberculosis. Cause tuberculosis. High lipid content. Then you should highlight it. That is put a Q. 17. That's a question related to that. Mycoric acid is responsible for staining characteristics. That's a slide you can study. And if it's acid-fast positive, what this means? It's a presumptive TB. You have to give the patients of antibiotics immediately. Okay, that's acid-fast stain. Okay, now in your mind, talk about mycobacterial tuberculosis. Then you think about when is the first time it mentioned? It's not here. The first time when we mention about Mycobacterial tuberculosis is back to exam 1, lecture 1. When we talk about the great scientist, which is Robocop, did a very important postulation. Don't forget, you remember he used guinea pig, but the sample here is tuberculosis. So when you do this question, when you see the tuberculosis, you know the basic knowledge, the background about it, but who is the first person really trying to do something for it is Bob Cook, and he created the postulate. So we have a question for them. Describe Cook's postulate. You can use your own words to describe. You don't have to recite or remember all those 4 things because let's say the first one is, you can say healthy animals absent from the microorganisms. Suspected microorganisms can be isolated to be a pure culture. Okay? Then inoculate will get the same disease. Finally is re-isolation. That's okay. Briefly is fine. As long as the key points is there. So when you look at that, you can think about the postulate of related to the mycobacterial tuberculosis. Okay, we solved this problem. Okay, let's see others, what we have. How about let's talk about those history. Okay. Let's look at these from the beginning. Okay, all cellular microorganisms go to one of 3 what? The domains. What are those domains? The bacteria, archaea, and eukarya. If you don't know, it's at the very beginning of the slides. So that means right here, you should highlight. That's related to that. Protists contain all of the following forms except. This is a little bit tricky because people doesn't really notice that when you have to read about here. Okay, so protists. You have algae, you have protozoan, you have slime modes. It's right here. What will be the answer? Fungi, probably not. Because you can understand about the fungi is a different categorization or different group compared to the protists. Who is the first to observe and accurately describe microorganisms? That's Dutch amateur scientist Van Gogh used his own mechanic microscope. The resolution power is 200. Who the following describe the evidence needed to discredit the concept of spontaneous generation? Rispasta, the swam neck. Remember the swam neck? Who is credited to developing and documenting the first vaccine against the smallpox? That's Edward Jenner. Remember the vaccine, what this means? VACCI indicate a cow. So the cowpox, the lesions can purify then against the smallpox. So Edward Jenner. The use of enrichment, I didn't talk about this. This is by Martinus Bellring. You don't have to worry about that. Fanny Hesse first suggested that agar to be used as solidified. That's true. Of course it is true. If you don't know, look at the picture here. So that's a question is very good. It's obvious. I don't talk about don't worry about it. Very simple question. What is a magnification? Magnification of objective lens, multiple ocular lens, 600. The answer is D. Okay, a microscope that exposes specimen to ultraviolet, blue light, and forms an image with light emitted at different wavelengths is called. So, what is it? This tells us The microscope uses light as an energy resource, but it's a different light. So you use blue light, violet, ultraviolet. What is that? Fluorescence microscope. If you don't know, let's say you forgot, that's fine. You go back. We're going to be looking for these slides. Then these are, we talk about phase contrast. Right here, what's the answer? Fluorescence microscope. So, once you do this question, you go back to read this slide and highlight the key points here. That's for you to study. So we're talking about fluorescence microscope. Let's talk about the last one. Confocal microscope, in combination with specialized computer software, can be used to create 3D images. Is that true or false? Yeah, true. It's have a 3D images. The reason is it will have to be used for observation of special matrix of bacteria. What's the name for that? Is biofilm. And we had a question for that. So these two, you can connect it together. Let me take a look. That's for it. Is that right? Let's go back to it. Do we have, I believe we had a question for the biofilm. Yes, we're looking here. So these two questions are connected. Complex microbial communities that grow tightly adhere to the surface are called biofilm. Then which tool we can do? Laser confocal microscope can do the observation. So two questions talk about one concept. Okay? So these we can connect it with each other. It's easy to do the review. Let's talk about something else. Resolution decreases when the wavelengths of the illuminated light decrease. Okay, that's something. If the wavelengths decrease, the resolution decreases. The resolution value decreases. Is that right? But the resolution capability is increased. Okay? So just want to let you know. So the answer is false. The resolution is not decreased. The resolution value is decreased, which means the capability is increased. If the objective lens of a microscope can be changed without losing focusing on the specimen, they call it what? Well, it's a very easy terminology we talk about in the lab as the first thing. What is that? Called a parfocal. That is why you use 10x objective lens to do the focusing. You move to 100x with all your immersion, only very limited adjustment. That's called a parfocal. As the magnification of a series of objective lens increased, the working distance decreased. Remember, in the first day of the lab, we draw on the blackboard. Prior, that's what we talked about. Okay, this one we talked about. These are we talked already. This is what we talked about. Let's do that. Because trans-electron microscope, question 19. Because transmission electron microscope use electrons rather than light, it is not necessary to stain biological specimen before observing it. This is the answer is true. In the electron microscope, we don't usually say it is staining. We say it's a coating. So it's a different. Okay, the terminology what we talk about is coating. Use heavy metal. We don't talk about staining. It's just the game of the wording. So we don't say that the other way, okay? Small intestinal cell structure can be visualized by. Now we mention about electron microscope. There are two different types, which is scanning electron microscope is look at the surface, 3D images, and transmission electron microscopes. We call it a transmission, which means it can penetrate inside, so it can see the internal cell structure. That's why it's called, the tool should be transmission electron microscope. So the answer is C. This is what we talk. Chemotaxis. What is the key we talk about chemotaxis? Is round trip. You go, you come back, like you go to the airport. So what we talk. So the answer is pretty obvious. Move toward an attractant or away from repellent. Now, let's say you don't know. That's fine, because this is not really, we talk about all the time. So you forgot about it, then let's look at here. This is it should be in the class 3. So we go here. Then my lecture 3 is very slow, so we come out. We didn't spend too much time talking about that, but this is what happened is chemotaxis. So you look at the chemotaxis is toward chemical attractants and away from chemical repellents. It's a round trip. Which of the following process can be used to bring nutrients into a cell against the concentration gradient? This is go back to talk about the nutrient intake by bacteria. We talk about passive diffusion, facilitated diffusion, active diffusion, and don't forget the group translocation. That is more important. But here talk about bring nutrients into a cell against the concentrate gradient. Here the answer is 2. Active transport and a facilitated diffusion. Is that right? Endospores are resistant to environment because calcium diproconic acids. Wait a second. The previous question is wrong. So, from high to low, yeah? The gradient facility is from high to low against, yes. Endospores are resistant to. Okay, go back to look at the endospore. What are they? The calcium, the diproconic acids. Calcium diproconic acids. They didn't mention about the very small acid soluble DNA protein, but that one is also important. Here only says calcium diproconic acid. Okay, that's the reason resistant to the environmental stress.
The answer to number 23 was active transport and facilitated transport.
Yeah, because it's against concentration gradient. Is that correct? No. It should be only A. Is that right? Yes. Yeah? So that's wrong. What's the difference between passive diffusion and facilitated diffusion? When you use carrier, when it's not, but the nutrients is always high to low. Is that right? Active is low to high. Okay, so you say, that's good. That means you studied. Okay, 25. You discover a new transport system used by a newly discovered bacterial species. The sugars that are transported using this system is phosphorated as they enter the bacterial cell. So which one is used this? This is not a difficult question. This is just using non-technical terminology to explain group translocation. So the answer is D, pretty straightforward. This is even easier than what we talk about in the classroom. Group translocation you can mention, you can look at here. Okay, we'll talk about again at exam 3. So when you see glucose, mannitol, glucose, they go inside. The phosphate has been transferred by different membrane systems and then become glucose 6 and the mannitol 1 because they can directly jumping into the glycolysis or modify the glycolysis status procedures and they can use more efficiently. So that's called a group translocation. This is Freud mosaic model 26, so everybody knows. So we just take it. Small circular DNA molecule capable of replicating and containing genes that are useful but are necessary to the bacteria is called what? It's a plasmids. So we say it's a linear, sometimes could be circular, most of the time more linear. It's about like 10, 20 base BP base pair. But the more important that we say it's could carry important information is antibiotic resistant. So bacteria, if they take it, what they call the transformation, is that right? The 3 methods for the communication genetics. So that's the plasmid. The cytoplasmic membrane of the bacteria. The retaining cytoplasm, its contents, act as a selective amine barrier. It's a major site for ATP synthesis. This is the answer is all of the choices. It should be not difficult. 29 we mentioned. Okay, periplasma space are true except it is found only in gram-positive bacteria. Of course, it's not. It is located between plasma membrane. Yeah, it's correct. It contains. So which one? Yeah, the A. It's found only in gram-positive bacteria is not correct. Bacterial ribosome is easy. 70S, very important. S stands for Vandenberg units. So 2 subunits, 30, 50, and 30. Okay, but they add up together is not 80. 80 is for fungi. You know about that. Bacteria is 70S. The movement of the molecules from the area of higher concentration to an area of low concentration without the participant of specific carrier molecules is called what? Higher to low. Passive diffusion, is that right? Permease proteins that is aiding the transport of nutrients resemble enzymes in their specificities for the substance to be transported. Each carrier is selected only a close related substance. The answer is true because at this moment the carrier is permease. We will talk later what this means. This means this enzyme. Whenever we talk about enzyme, which means it's very specific, it's a group or the similar or the same substrate. Okay, so that's a little overhead, but it's not really difficult. So that's true. 34, we talked. This question we haven't talked about that. You've identified the above gram in characteristics of a particular cell type. So we already know it's gram-negative. How would you modify the diagram to depict the other cell type and it was discussed in the class? The only 2 modifications. What are the only 2 modifications? This is going to become heavy. What else? You can say you took out that part. You can say lipopolysaccharides become teichoic acids. Either are fine. Okay, let's talk about these are the medias. 36 is relatively straightforward. Medium containing some ingredients of unknown chemical composition is called. That's called a complex media. Is that right? It's not defined media. Defined media and synthetic media basically here are the same, which means every chemistry ingredients has to be well defined. So the answer here is B, complex. All fastidious microorganisms require which of the following to grow? So we talk about in the lab already. We're using a very special media, looks like a chocolate. It's a chocolate-like agar to grow fastidious bacteria. So the answer here is chocolate agar. But we don't have it. The answer is extra nutrients because chocolate agar contains extra nutrients. Okay. 39. You'd love to culture bacteria that grow in the lung of a human patient with pneumonia. Which of the following bacterial media you should use? This is a little bit ahead of time, but we should be know. Remember, I said it's a very important slides. You should definitely know, and we will talk about it again, which is right here. Which agar plates is a blood agar. A blood agar, you could see Streptococcus pyogenes, Streptococcus pneumonia. Both of those are lying infectious disease. We call it upper respiratory tract infectious disease because we get to know alpha or beta hemolytic. At the same time, it tells you blood agar is a differentiated media. So here, complex media supplemented with whole blood. That is a transferase term talk about blood agar. Blood agar is both a differentiated enriched media. Yes, it's true. Most bacterial cells divided by binary fission. Is that right? 1 become 2, 2 become 4. That is what our quiz at the beginning, the math talks about. That's why so 42 is, we already talked about. What is this called? Generation time. So 41, 42, and 43 plus bacterial growth curve, you should put them all together to study. And these 3 questions are really good for you to understanding the generation time and the growth curve. Which of these methods can be used to determine the number of viable microorganisms? Okay, that is talk about how we isolation of bacteria. Light scanning color spectrometer, measuring total cell mass. Those we can know the bacterial density, but we don't know whether they're live or dead. Measuring CFU, colony forming unit, that we know the live cell. We will practice spread plating and pore plating next week. So Thursday no lab. Next week we will do spread plating and pore plating. So we get to know how to calculate colony forming units. Counting a known volume of cells in a hemocentimeter, that is, we don't know the live or dead. Okay? Now, all these questions here, 45, 46, and 47, this is basically something probably you have to memorize a little bit. That's not many tricky things. Organisms grow well at 0 and have optimal growth temperature 15 degrees Celsius. That is called sacrifice. If you don't know, you go back here. This is not many strategy. Okay, that's sacrifice. Organisms that do not require oxygen for growth will grow better in the presence, what is called? Facultative, yes. Facultative anaerobes. Most microorganisms maintain its internal pH. Well, near neutral pH. This is the one, I don't think it's a very good question. Because A and B kind of also makes sense a little bit. It depends what type of the bacteria. So the best answer is A, is near neutral. But the year, if you pick near the optimal growth pH, I still give you the points. So A or B is fine, but better is A. Because the best answer is A. 48 is fine. We already talked. 49. Organisms that ignore oxygen that grow equally well. Allotorrent. Grow equally well with oxygen. When grow blood agar clear zone of Streptococcus pyogenes, that's beta hemolytic. We will mention real quick Streptococcus pyogenes. The energy required to transport with ABC transporter, that is ATP binding cassette, is derived from what? Protein ingredient, no. Electron charge separation across the membrane, ATP hydrolysis, a simple mechanism. It's called I didn't mention about it. It's coming from the ATP hydrolysis. Okay, it's a little deeper than this question. This is a relatively good one. Sample from the lake. We do pore plating and spread plating. And we find that fewer colonies on pore plating than spread plating wire. Because pore plating using melted agar, they mild heat. You will feel about next week Thursday. I will ask you to do more quickly, because if you do it slow, it's cooling down. It's all going to be solidified in the bottle. But what happens when you pour real quick, they'll mild heat. If it's very heat sensitive bacteria, it will be dead. So the answer here is heat sensitive microbes will kill. Resistant to penicillin, 53. Why? Very simple. Because bacteria containing all plasmid, is that right? The all plasmid carrying the information for antibiotic penicillin. Can we do that? This should be easy. Which culture has the longest lag time? Culture 1. Faster growth? Culture 2. Shortest doubling time, also culture 2. Highest cell density? Culture 3. That's easy. Okay, what are the two bacteria has endospore? Clostridium and Bacillus. They will have the endospore. Okay. What's the function of the endospore? Is protection in the stress environments. Remember, in the lab we say it's a protection structure. It's not a reproductive structure. So mother cell has does not mean daughter cell will automatically have. What is magnification and resolution? Magnification is the enlarged. Resolution is the capability to differentiate two closely attached particles. What is this one? 57? Monotrichos. Once you learn monotrichos, at the same time, you should go back at your slides. I say this all the time. You should study this. Because patterns of the flagella distribution is monotrichos, antitrichos, levotrichos, and the petritrichos. We have a corresponding picture for that. Okay? It's very simple. The last one. I want to say this. Exam 1, basically we say it is straightforward. I would not say easy. Because you know it's easy. If you don't know, it's difficult. Basically, we say, compared to exam 2 and 3, it is more straightforward. So if you focus on the practice exam questions, look at your slides, like what I said, you highlighted, you should be doing pretty good. That's what our previous 10 years experience is. Okay, so you don't have to be panicked about that, but you do have to study. Okay.